Okay, so in the last video I had completed the nerve portion of the unit 1, and this video I will start with the muscle. So in muscle, in your syllabus you have ultrastructure of skeletal muscle and molecular and chemical basis of muscle contraction. Okay, so let's start with muscle and its types. So muscle, as you already know, that it is a tissue that is composed of cells which have the contractile ability, like they have the contraction and relaxation ability, and they are mainly useful for the movement, support, heat generation, blood circulation in our body. So the muscle tissue consists of bundles of elongated muscle cells, and all these muscle cells are richly supplied with blood vessels. Also, muscle cells can be of three types: skeletal muscle, smooth muscles, cardiac muscle, depending on the location and also their structure. So in skeletal muscle, if you can see that the fibers are striated, which means it has stripes or striations, and they are tubular in shape and also multinucleated. Okay, there is more than one nucleus in one fiber. Also, these muscles are voluntary, which means that you can control their function or action according to your own will. Okay, and skeletal muscle, because they are attached to the skeleton, hence the name. Now next is the smooth muscle, which means they do not have any striations. They are non-striated muscles. The spindle shape, which means middle portion is wider and tapered towards both the ends. And uninucleated. Unlike the skeletal muscle, smooth muscle fibers are single nucleated cells. Okay, and involuntary, because you cannot control the functioning of the smooth muscle, and also it's present in the walls of internal organs. And in case of cardiac muscle, the fibers are striated, branched, as you can see that it is forming branches here. And also cardiac muscles are involuntary because we cannot control the heart muscles on our own, and it is present in the covering of the heart. Okay. Now this is skeletal muscle. So basically the skeletal muscle remains attached to the skeletal system, that is the bones and all, with the help of a connective tissue known as tendon. So tendon is the connecting point between a bone and a muscle. Now, skeletal muscle is the largest muscle tissue in the body, and it is almost around 40% of the total body weight. So if you can see that it is swollen in the middle and tapered towards the end. Okay, and each muscle consists of thousands of muscle cells or muscle fibers. Now each fiber can be long enough to cover the entire length of the muscle, or it might be shorter than the total length of the muscle. Okay, different types of coverings are present here. The first covering, which covers the total bundle of the entire muscle, that is known as epimysium. Okay, inside the epimysium there is a middle layer surrounding each bundles. You can see each of these bundles are called fasciculi. Okay, now each bundle is covered by another layer which is known as the perimysium. Okay, now if you can find inside the fascicle, there are individual muscle fibers. Okay, each individual muscle fibers are covered by another layer which is known as endomysium. Now each individual muscle fiber is, if you can see it in enlarged view, is covered by a sarcolemma, which is the plasma membrane of the muscle cell. So it's a sarcolemma, and it encloses the cytoplasm inside the fiber, which is known as sarcoplasm. Okay, and inside this sarcoplasm numerous myofibrils are embedded. Okay, so and because it is why is it multinucleated? Because it is formed by the fusion of several myoblast cells together. So all those nuclei of all those cells have remained intact, and therefore it is giving off a multinucleated or it is known as sinusitic structure, and this kind of structure is known as syncytium. Okay, where there is a multinucleated condition. So this is the basic structure of the skeletal muscle. Now moving on to the ultrastructure of the skeletal muscle. So this is basically a muscle fiber. which is covered by sarcolemma, and you can see that there are numerous mitochondria inside this muscle fiber, and each of these rod-like thin filaments is known as a myofibril. Okay, so embedded inside the sarcoplasm, that is the cytoplasm of the muscle cells. Now in this muscle fiber, you will see that there is striations, like there is a light band and there is a dark band. So light and dark, there is alterations of light and dark bands throughout the length of the muscle fiber. Okay, and there are multinucleated, since this is a multinucleated cell, so you will find that nucleus number is many. Okay, so if you consider a single myofibril and if it is enlarged like this, you will see that there are alternate light and dark bands. So the light colored bands are known as I band or isotropic band, and the dark colored band is known as A band or anisotropic band. Okay, so if you can see here that this line is called a Z line or Z disc, and this is also another Z disc. So from one Z line to another Z line, this unit is known as a sarcomere. So throughout the length of the myofibril, there is basic repeating units of sarcomere, which is the region between two Z lines, and it consists of thick filaments, which means these thick red colored lines, and thin filaments, which means this green thin lines interdigitated together. That means they are forming a meshwork like structure, and this kind of repeating unit is known as sarcomere. Okay, so these thick and thin filaments are basically responsible for the muscular contraction, and thick filaments are made of myosin protein and the thin filaments are made of actin protein. Okay, now thick myosin filaments, they are arranged parallel to each other, as you can see here, and it is forming the dark band, also known as A band or anisotropic band. Okay, and actin and myosin are together known as myofilaments. Since these are the two main filaments of a myofibril, so they are called myofilaments. Now, if you can find in this region, there is no thick filament, only light band is there. This light band is known as I band or isotropic band. Why is it anisotropic or isotropic? Because when polarized light is passed through them, then the light rays are reflected in different directions in case of anisotropic band, and the light rays are reflected in only one direction in case of isotropic band. That's why the name, but you do not have to go through those details, but just like remember the namings I band and A band. Okay, now the thick myosin filaments, they have a central portion which inside the dark band, which is slightly lighter than the two sides, because there is no thin filament here. Okay, and in the two sides of the A band, you will see that thin actin filaments and thick myosin filaments, they are together running parallel to each other, therefore giving a more darker color. So if you can see in the central part, there is no thin filament. So the central part is relatively slightly lighter than the two sides. This part of the A band, center of the A band, is known as H zone. Okay, or Hansen zone. And through the H zone, a line runs straight, which is known as the M line, because it is running through the middle of the A band, and it is also this M line marks the center point of each sarcomere. Okay, so here, so this is the A band, which is the length of the myosin filament. This is the A band. Now the I bands are the thin filaments running in the light band. So this is the I band. Now thin filament, where in the regions where it overlaps though excluding those regions, this part is called the H zone. And what are the Z lines? Each individual sarcomere is flanked by dense protein discs called Z lines, from where the thin actin filaments radiate out on either side. So you will see that Z line is basically the center of the radiation of the actin filaments. So actin filaments basically radiate out from this part. Okay. So next is the structure of actin. So actin is a very well-known protein. It usually exists in two forms. One is the globular G-actin and the filamentous F-actin. So in case of G-actin, it is a monomer, that is the subunit of the actin protein, and it is forming the basically forming the filamentous F-actin. So this monomeric G-actin, when it is bound to ATP, it is known as activated, and therefore it forms stable dimers or trimers, and then it goes on to assemble into a filament, and it is known as F-actin. So this ATP is hydrolyzed to ATP and inorganic phosphate PI, and then G-actin polymerizes to form actin. So you can see that in one end of the filament there is assembly of G-actin, and in another end there is disassembly of G-actin, and this disassembly is mainly of ADP bound G-actin, not ATP. So these two filaments are formed and they twist together to form a microfilament strand. Now the two ends of the actin filament are quite different, because here assembly is happening, polymerization is taking place, here disassembly or depolymerization is taking place. So because of that, this end is known as a plus end, and it is also called the barbed end, where there is faster polymerization, and this end is called the minus end, and it is also known as the pointed end, where the depolymerization is taking place. So polymerization is easier at the positive side and difficult at the negative end. Okay, so this dynamic assembly and disassembly of G-actin and helping in the polymerization and depolymerization of the actin filament, this is causing what we know as the actin treadmilling. Like from this side it is adding up the units, and from here the subunits are getting disassembled. So this is known as actin treadmilling. Okay.